Michael N.A. Eskin has made exceptional contributions to research on edible oils, particularly canola oil, for which he received many prestigious awards, including the Order of Canada in 2016. He is one of the world's leading food science writers with 17 books to his credit. His book, Biochemistry of Foods, now in its third edition, is used in university courses around the world and has been translated into several different languages.
Good morning. My name is Michael Eskin, and I'm a professor at the University of Manitoba in the Department of Food and Human Nutritional Sciences. My short presentation today is titled CANOLOL, an Emerging Bioactive. This presentation is dedicated to the memory of a very close colleague, Dr. Usha Thiam Hollander, with whom I had the privilege to work for the past decade. She was an amazing young woman who passed away tragically from cancer on December the 24th of 2020. As a result, I took over the supervision of most of her graduate students. In the next slide, phenolic compounds are the subject of extensive studies worldwide because of their antioxidant and anti-cancer properties. Our group conducted research on canola as it is one of the richest sources of phenolic compounds. This presentation is focused on the extraction and utilization of phenolics from canola and mustard, with particular attention to the formation of Canalol. Canola is the third largest global oilseed after palm and soybean oils. The annual production is for the oil is 4.5 and the meal is 5.79 million metric tons annually. And I'm working on mustard, which I've worked before on identifying unique mucilage. And while the mustard production is much smaller, it's still, Canada is nevertheless one of the important producers of mustard seed for the mustard condiment industry. In the next slide, the predominant phenolic compound in canola is sinapine, which is a choline ester of synabic acid. And this accounts for about 80% of the total phenols, while synabic acid is the major free phenolic acid. The presence of Canalol, which is 2,6-dimethoxy-4-vinyl phenol or vinyl syringol in crude rapeseed, was first discovered and characterized in Japan by Koski in 2003. They were using a low erucic acid oil. The high level of phenolics in the post-expelled oil was shown to correlate with its antioxidant activity, and the formation starts with the removal of choline from synapine to form synabic acid, and then the decarboxylation of the synabic acid to form canolol. Of these phenols, canolol is the most active antioxidant, and its formation is really quite important. In fact, Wakamatsu and co-workers in 2005, also in Japan, confirmed the presence of canolol in canola oil and showed its superior antioxidant activity over such antioxidants as alpha-tocopherol, vitamin C, beta-carotene, rutin, and quercetin. In the next slide, You see the processing of canola, and the byproduct is produced, which is canola meal. And this is still highly underutilized source, and it's used mainly for animal feed. It is very rich source of synapine and synapic acid. The challenge was to catalyze its conversion to Canalol, and we examined different pre-treatments to accelerate the formation of Canalol, followed by environmentally green extractions. The traditional method for obtaining canola oil involves the extraction of the oil with hexane, and then the removal of hexane and leaving behind the canola meal. This byproduct, as I mentioned, is limited to animal feed, although certain protein fractions are now being commercially separated for use in foods as ingredients. Using the high pressure and temperature processing, Canalol was shown to be structurally transformed from synapic acid via decarboxylation to Canalol. And using this accelerated solvent extraction system, ASE, using it at high pressure, 1500 psi at 160 degrees centigrade, canola meal was shown to produce the highest yields of Canalol, using 70% ethanol as the green extractant. This clearly established ASE as an efficient green method for extracting phenolic compounds such as Canalol from canola meal. We just published a paper using air frying as a roasting pre-treatment for enhancing the recovery of lipophilic synapase, including Canalol, from various mustard samples. And we were looking at lower grade yellow mustard seed, and we were able to produce high yields of Canalol compared to Oriental and the higher grade yellow mustard samples. And certainly that indicates the potential economic benefits from even a low yellow mustard sample. In the next slide, synapates, particularly Canalol, is of great interest as it has proven to be a powerful antioxidant and anti-cancer agent. In our laboratory, we showed that crude Canalol and canola distillate extracts improve the stability of refined canola oil during deep fat frying over 5 days for a total of 30 hours. And the red cone really indicates the Canalol. As you can see, it's really more effective than BHT. And DDL would be a crude extract containing Canalol. And this has certainly been corroborated by other studies showing that Canalol is effective in enhancing the stability of high oleic canola oil by Mathias and co-workers in 2014. And using a pressurized solvent extraction system with alkaline methanol at 1500 psi and 100 degrees for 5 minutes by Lee and Guo in 2018, they showed that it was as effective in producing Canalol that compared to using soxhlet extraction at 100 degrees for 12 hours. A similar pattern was obtained for both treatments in terms of the phenol content and the antioxidant activity, so that as expressed by DPA, the DPH value, so that one didn't have to use soxhlet, which is certainly impractical but certainly the pressurized solvent extraction was very effective. In the next slide, the studies have also demonstrated canolol's important anti-cancer properties, both sinapine and canolol. Jiang, in the study on the in the top left-hand corner, he examined the effect of canolol on human gastric carcinoma cells, and you can see it found to cause apoptosis, increasing concentration, compared to the normal human gastric mucosal cells. There were very small changes. In fact, it was considered to be non-toxic at the levels used to destroy the carcinoma cells. Another more recent study by Yang, this looked at the effect of sinapine to reduce melanin synthesis in ACE375 melanocytes by inhibiting melanin production and by anti-tyrosinase activity. And you can see there's pretty effective increase in inhibition, which suggested its potential as a whitening agent. Further research on canolol, which we are continuing in our laboratory in collaboration with colleagues at Laval University, plan to pursue some of its anti-cancer properties. Thank you very much for your attention.